Hi, everyone. Welcome to the 36th session of the MedAI group exchange sessions. And this week we have Bin Lee from the University of Wisconsin-Madison here with us to present his research on weakly supervised tumor detection in whole slide images. So Bin is a PhD candidate in biomedical engineering at the University of Wisconsin-Madison, and he is currently a research assistant in the laboratory for optical and computational instrumentation, mentored by Professor Kevin Eliseri. I don't know if I'm correct pronouncing it correctly, Kevin, but who's also here with us today. And he develops computational methods to improve the understanding of biological and pathological mechanisms of disease development and patient care based on multimodal microscopic image analysis. So thanks so much, Bin and Kevin, for joining us today. Before we get started, do you have any preferences on how you'd like to take questions today? Are you okay with us interrupting you, or would you prefer it at the end?
I think either way is gonna be fine. So yeah.
Awesome. Okay, so let's try to make this session as interactive as possible. And without further ado, let me hand it over to Ben.
Yep. Sounds good. Okay, I think we can get it started. So my name is Bin Lee, and I am a PhD candidate in biomedical engineering. And first of all, thanks for inviting me today. I'm excited to be able to present my work here. So today I'm going to discuss with you the topic of weakly supervised tumor detection for whole slide image analysis, and cover one of my recent work on building deep neural nets for whole slide image classification. So at the beginning, I want to give a brief introduction on histopathology and its role in clinical routines. Histopathology usually involves tissue extractions through the procedure of needle aspiration or resection surgery. And once the tissue are extracted from the patient, it usually gets sliced, stained, and fixed onto glass slides. And this procedure is usually referred as biopsy preparation. And once those biopsies are ready, they are transferred under a light microscope and examined by medical experts such as pathologists. The pathologists will look for the histomorphological features of the tissue microenvironment, as well as the cellular level features, for example, like cell types and shape, and the cell organization and the glands and ducts organization. And they are usually looking for the trace of metastasis and the fibrosis to match the criteria of diagnosis. So you are probably more familiar with the medical imaging modalities, such as the CT scanning, MRI scan, and ultrasound. Unlike those medical imaging modalities, often look at the mainly look at the organ level features of the patients. So by contrast, in histopathology, the goal is to examine the tissue features in a microscopy level, basically the cellular level, and usually this histopathology is the standard way to ensure the accurate diagnosis for the cases that the medical imaging modalities are not able to tell exactly what the disease is. And also it's the gold standard for most differential cancer diagnosis, such as the sometimes the chronic inflammation versus cancer, which can usually be confused in a radiology level. But overall, the tissue extraction is usually guided by medical imaging. So basically, the approximate location of the lesions are usually first determined by the medical imaging modalities, such as ultrasound, and then the tissue of the potential lesion regions are then extracted and examined. So in the 1919s, commercial whole slice scanners emerged and have become one of the core tools in modern pathology. So this type of scanners are able to convert glass slides into digital images that has a very high resolution. So basically, they're able to scan the slides in multi resolutions, and then once the images are scanned, then the pathologist will have the ability to look at the slides and zoom in and zoom out, just as if they are looking at a real physical slides. But all of these are stored in the digital copies. And since because all of those features are preserved in a microscopic level, and the tissue section can usually have like several centimeters by several centimeters in the physical slides. So that means that for each slide, the image can have like several gigas of pixels, and that is also one of the challenge that involved in most of the analysis tasks. And compared to radiology, this is relatively a newer field, and there has been a lot of open questions and challenges that remain to be solved. For example, like some of the emerging applications in computational histopathology, including automatic tissue segmentation and slide screening and retrieval. This is usually referred to that when the pathologist is given a new slide at hand, they usually want to search through the database for the most similar patients so that they can have a better idea of how this patient will going to respond to a specific treatment, or whether this is actually an accurate diagnosis or not. And as you can imagine, before the digital scanners, this is almost a really difficult task, because then that means you have to search through some physical storage of the slides, and usually it's just impossible to do. So so that is like one of the major advances brought by the scanning techniques. And also move beyond that, there are currently a couple of applications that are associated with how to correlate the image-based feature with the genotype. And this is usually a quite difficult task for the pathologists to do. But given the advances in neural nets, and now we're able to process those images in big batches, on a very large scale, then this correlation is shown to be promising to be found. And also, for example, another very popular application is the survival analysis. So basically, this is also quite a challenging task just based on the image features. For example, based on the traditional examination of the medical experts. But since now there are a lot of architectures, computation models that allows you to basically correlate the image features with the survival data of the patients. Now it has been shown that there is some of like very interesting and important features in the images that are usually overlooked by in the traditional examining process. But now it's been shown that the machine has some capability to find out those features, and that's exciting. So in the following presentation, I will focus on one of the essential problems in whole slide image analysis, which is the whole slide image classification and tumor detection in whole slide images. So perhaps one of the biggest challenge of analyzing these images is the vastly high resolution of those images. So for a typical whole slide image, it can easily have up to 100K by 100K pixels. And as you can see, this is just too big for convolutional neural networks to handle at once. So you will never want to fit this whole thing into a neural networks. That is just impossible to do. And the other challenge of this is that localized annotations are hard to obtain. As you can imagine, if you were to draw these cellular level annotations on the whole slide images, that would just require a lot of effort. And also drawing annotations is just not a part of the clinical routine. So usually this is being all of the slides that most of the slides in the clinics are usually just labeled as malignant or not. So they usually just have some global level information about how the slides contains the tumor or not. So that means a lot of the regions can naturally be normal tissue in a malignant sample. Although the image that I show here actually has annotation, but this is one cured data set for an open challenge. So, but in the real case, this is usually not localized annotations are usually not associated with not come with the slides. And from a computational perspective, a standard practice is to crop the host images into small patches for analysis and detection localizations. So I would then try to introduce some of the related work on this and the previous work. So we first take a look at a scenario of patch-based classification. So if we have given some normal patches and some patches with cancer, so this is a straightforward question, straightforward test. So we basically extract some image features out of it and then fit it into some sort of classifier, and which is supervised by some patch level labels, and everything you can just treat it as a standard image classification problem. But the issue here is that we must have the labels for all of the patches extracted from host images. So just give you a number here. If you are trying to label those patches, you can easily get more than like 10,000 patches for each slice. And that is just the same question as, okay, if you just almost as label intensive as you were trying to draw cellular level annotations on those images. So, so we ask this question, what if we don't have these patch level labels? If we only know whether the slice contains tumor, can we still try to train a model that does everything, does the job that we wanted it to do? Given that we can actually gather a lot of these unannotated slides with the global level labels, we might still be able to do it. So, so that is because most of the slides in the clinics are annotated, and the malignant sample basically means that it contains malignant regions. And also, as I mentioned before, drawing an annotation is difficult. So there might be way that we can still build a model on the patch-based solution, but it's only supervised by the global level labels. So let's take a look at this scenario. If we have, for example, a slide that is labeled as malignant, and we wanted to extract some patches from it, there will be some normal patches. There will be some patches with cancer, but we do not know about that. So basically, we get a mixture of positive and negative samples from a positive slide. And if we extract, by contrast, if we extract patches from a normal slice, then we were sure that all of the patches are negative, and otherwise it would be just labeled as a malignant slice instead of a normal one. So, so as we can see, this is different from a standard supervised learning problem because the label only indicates the existence of a positive instance in a set of images instead of each individual image. So this is actually a quite classic image classification problem called image set classification. So now, so in this scenario, those each slice are treated as a loose set of images, And we have some sort of classifier that we wish that it could give us some predictions on each of the individual patches and supervised by global level labels. And usually those images, those patches in the slides extracted from the slides are called instances. And each slide is then called a bag, which contains all of the patches. And this problem is known as multiple instance learning. And noted that this is actually an oversimplified assumption of the real task here, because we treat the patches extracted from whole slide images as the loose set of images without considering the spatial relations. But in the following presentation, I will show that although this is an oversimplified assumption, it can work in a lot of real world cases actually. And then I will also discuss what is the shortcoming of using this oversimplified assumption and potential solutions for that. So, and another motivation of why we cared about using weakly supervised method at all, like you can imagine maybe it's just better to have, like in terms of going for a higher accuracy, maybe you can ask people to annotate the slides, but the label is worthy because we have a model that have a higher accuracy. So in this study basically shows that so the figures, the performance, the figure that shows from this study shows that a weakly supervised model on a large data sets will lead to a higher generalization performance than the fully supervised learning model on a smaller curated data set. So basically what this figure shows is that, so this figure, this data, so let's just take a look at the figure B. So basically the MSK is a weakly supervised, it's a data set used for training a weakly supervised model, and a Chameleon 16 is a fully supervised model. So if we test the weakly supervised trained model on MSK dataset directly on a Chameleon 16, which has some sort of domain shift, but some of the domain shift, but quite similar data, we see that there is a drop of about 7%. But if we do the other way around, we use the fully supervised model trained on a Chameleon 16, which has significantly small number of total slides, and then test that directly on the MSK dataset, which are not annotated, but has a significantly more number of slides, then we see a really big performance drop here. So although we have in the fully supervised training, in the fully supervised dataset, we have a lot of labeled patches, like more than like 1 million labeled patches, but it does not generalize good on a third part data set. But if we train on the other way around, if we use that weakly supervised model trained on that big data set that has a lot of annotated slides, it can perform decently on a third party data set. Okay, that is one of the motivations. So in practice, practically speaking, this will be the method that is preferred in order to achieve some real world application. So let's then take a look at this standard multiple instance learning assumptions. So basically, it's assumed that in the back we have a bunch of instances, and each instance has a hidden label that in the binary case, it belongs to, it takes a value of 0 or 1, but we do not know exactly what the labels are, but we know that they exist. And the label of the bag is then given by these equations here. So basically, this is just saying that it equals to the bag label equals 0 if all of the instances have the label of 0, and it equals 1 if at least there is one instance that takes the label of 1. So, and suppose there are some suitable functions G and F. The the bag labels can be decomposed into this form. So basically, it's we have some unary function f that operates on the instance, and then we have an aggregator G that operates all of the outputs of the feature extractor. And then if due to that we do not have any specific assumption of the order of the instances or any spatial relations of the image patches. So the G has to be a permutation invariant. And some popular choices are max pooling function and some attention-based pooling functions. And very recently, the visual transformer, which is self-attention. So let's take a look at the baseline solution with max pooling. So in max pooling, we use a max pooling operator that operates on the bag level. So this max pooling operator will only basically select. So basically in the positive bag, it will select the instances with the highest probability to be positive. Basically has a higher score to be positive. And for a negative bag, it also select the one with the higher score, but in that scope, it's basically saying that select the instances that are less likely to be negative, right? So if you ensure, so in the negative bag, if you ensure that the instance that is less likely to be negative, it's still negative, then all of the instances that are more likely to be negative will of course be negative. And for the positive bag, if you select the most positive instances in the bag and ensure that that one is positive, then this will overall ensure that within that bag, at least one instance will be positive. So that is why the max pooling can work in this way. But as you can see that there is no guarantee that you can, one problem with the solution is that there's no guarantee that the decision boundary will exactly tell you, exactly delineate all of the positive instances in your positive bags, because it will of course get shifted towards the positive space, because we're only looking at one pattern in the bags that matters. So yeah, that is one problem, which means that we need some further constraints to delineate the positive instances that are less positive. So another, in the real practice with whole extra image processing, another problem with the solution is that suppose we have this kind of workflow that we have some classifier that give us some scores from each image patches. And this actually, no weight updates happening in the whole process until we actually select the one with the maximum probability, with the maximum output score. And then we basically the weight update only happening after we select the max, the instance with the maximum score. So this means that if we use like CNN as a feature extractor, it will get, it would suffer from insufficient training because it was seeing so few instances for updating the gradients. And another issue is that suppose your positive bags are actually dominated with negative samples. So you have really small tumor regions in the whole slide. So basically the fraction of positive patches, it's super small in your positive slides. Then because the very nature of the training at the beginning, it's basically trying to select some random patches. And then which means that the gradients can be actually very noisy at the beginning. And sometimes it will take really long time to converge. And so that is why there's another categories of methods of multiple instance learning that involves the learning of a back level embedding from the instances. So we first have some add function that learn the feature extractor that learns some representations from each instances. And then we have some function aggregator function G that first learn a back representations out from it, and then have a classification here that predict the back label. So this gives us more flexibility and also as you can imagine, potentially the gradients can actually flow, the supervision signal can flow through all of your feature extractor in this circumstances. And also due to the assumption G has to be permutation invariant. So for example, one of the selection will be, you can do some sort of like soft selection of the features. So basically like attention mechanism. And also this learns a joint representation of all of the instances in the back and usually leads to more accurate performance. So that's actually one of the work that actually use this kind of ideas. So basically, they're trying to use some attention pooling to solve this multiple instance learning problem. So you have set of attention block, which is a set of neural networks that learns the attention score from each instance, and this attention score then is multiple. Then it's multiplied with the corresponding instance level embeddings, and then you will basically get a back embedding that is a weighted sum of the instances embeddings, with the attention scores being learned from the neural network, basically multi-layer perception, and this means that then you just train the network end to end, and you wish that positive instance will eventually have high attention scores. And it turns out it's actually the case. And the another solution will be use the more complicated gated attention block that actually also does a selection in along the feature of each individual instance. But the idea is similar. So, but the problem with this, although this has some quite nice performance, but it does not actually directly model the instance to instance relation. Instead, it only learns the joint representation of the whole bag. So that is why a more complicated categories of solutions involves the learning of involves another type of powerful structure with neural nets, trying to basically allow the features to be updated based on the information of other features. So basically you can let information flow between the instances. So for example, one popular choice is the non-local networks. So a non-local network takes a general form like this. So for example, in the image, the instances xi here can be some sort of the local features extracted at different locations. And the non-local operation updates its feature by aggregating all of the information from other locations transformed by the unary function g, and then weighted by some sort of pairwise function f that takes in the feature at that location and the other locations at the input. So so this might be more familiar, might seem more familiar to you. So if you choose, like one of the example of non-local operation is self-attention. So basically, if your aggregation function takes this form, then it's just self-attention. So you have basically your pairwise function f here is basically that they basically compute that self-attention matrix for you, and then you weight it. Then you update the features by using the weights from the weighted sum from all of the other inputs, using the weights that is computed in this self-attention matrix. So when it's used in multiple instance learning, so this means that each instance can now correlate it with other instances. And the updated instance embeddings can potentially contain information of the whole bag. And also self-attention is permutation invariant. So it's it obeys the assumption there. But the one problem with this is that although you might have fancier features now, a pooling operator is still required. We can take a look at what at the actual case here. So we fit all of the patches into some sort of embedder that gives us embeddings, and then we feed the embeddings into self-attention model, like it can be any kind of transformer, and that will give us another set of features that are updated and potentially have information from all of the other embeddings. But we still have to aggregate them. We still have the read. Basically, we still have to have a readout layer sort of thing. So what do we choose for the aggregation function here, so that we can actually supervise it by the back level prediction? So our idea is, the idea here is to design a pooling function using the idea of self-attention and the non-local operation. But now we consider correlation between the instances and produce a back embedding with a constant size, just as the attention-based pooling. So we propose this following aggregation function called dual stream multiple instance learning pooling. So all of the H here are the embeddings that is computed from the patches. So in the first branch, we have a standard max pooling operation. So basically, we determine the instances with the highest score to be positive. And then in the second branch, after we found out that instances, we call that instance a critical instance. After we find the critical instance, we then compute we then use the idea of self-attention, like we transform the embeddings into the query and the value vectors. So basically, we have two vectors that are transferred by two separate sets of neural nets, and then we compute a pairwise similarity between them. So basically, we use the Q vectors to compute the similarity, and that is done by an inner product. And then basically, what we're trying to do here is that we compute the similarity between the critical instance to all of the other instances, and we use that as the attention score, use that as the weight to in the second branch. We learn these back representations by summing up all of the value functions, which are also transformed from the embeddings. But we use the weights, which is computed by which is determined by the similarity of all of the instances to that critical instance that we pulled out from the max pooling. So this means that instead of just using a normal attention mechanism here, we actually use regularize this process by using a trainable measurements between the critical instance to all of the other instances. And this is also permutation invariant, because the max pooling is permutation invariant, and in the second branch, we're just basically computing the pairwise similarity, and which is also permutation invariant. And so this might sound a little bit like a graph model here, but there is actually a fundamental difference between there is actually important difference between a graph model and a non-local operation. So in the graph model, basically when we update the features in the nodes, we basically sum up all of the features. We aggregate the features from all of the connected nodes, and the features from nodes are transferred by a set of learnable weights. But in the non-local operation, the vertices are connected with the functions of the two vertices that are being connected. So this actually has the sort of like, for example, if you use similarity measurement, then this is there is a kind of regularization here. So this actually also intuitively this will bring better. This will result in better decision boundary for the instance level classification, because now we have some sort of soft constraint on the decision boundary that is being learned for the instances, because now we are having some a separate trainable measurement that actually select all of the instances that are less positive if you're purely using a max pooling mechanism. And this model can also easily handle the multi-class multiple instance learning problem. So basically, you can imagine that for multi-class multiple instance learning problem, we can have multiple max pooling branch. So we can pull with respect to each class. And for each class, we will determine a critical instance. And then we can, then our back embedding will have more entries, and the number of entries will equal to the number of class here. So basically, we compute different sets of weighted sum, and each set of weights is computed by using that critical instances of that class, the similarity of the in critical instances of that class to the rest of the instances, including that instance itself. So yeah, and then the last fully connected layer then we'll have a channel number equal to the number of class, and of course in this case you will have to use a distributed encoded class label. So you basically have a one-hot encoding label as a vector as your labels. And then we take a look at the training process. So if we trying to train the whole thing end to end. So if we don't have a lot of image patches, this is good to go. We can basically have patches that each patch is processed by a CNN, then the CNN produces the embeddings, and the embeddings are then fit into the aggregator. But this is okay when we don't have a lot of image patches. As you can imagine, if we have a lot of image patches, then that means that you will have to keep all of the image patches in your memory. Although we are not trying to use a CNN or a super big image, we are just trying to use each small CNN on each individual small patches, but we still have to put all of the patches into the memory. And this become unrealistic if we're trying to process some real world data set. So this is the problem that I just described. So this requires, although this aggregator has a potentially better performance, but it will require the whole bag to be fit into the memory at a time. And also the problem of noisy gradient can still exist, because whether it's attention-based pooling or the max pooling, if your data if your data set is really unbalanced in the positive bags, where each positive bags only have a small fraction of positive instances and the majority of the bags are still negative, then the training can take a long time to converge. Like for example, like this image that I show here. So that is why we utilize another technique to pre-train our feature extractors. So we use contrastive learning, self-supervised contrastive learning here. So so we just treat each image patches as an individual image and feed into this sync layer, which is one of the very popular self-supervised training pipeline. So we basically just directly use that almost. And then after this, well, and another nice thing about it is that we can actually generate a lot of unlabeled patches from those images. So which potentially will give us better features, better self-supervised features. And then after the feature extractor is trained, the multiple instance learning networks, the aggregator only observes the pre-computed embeddings, and then it gets trained in a separate phase. The multiple instance aggregator is then trained in a separate phase. And in some circumstances, this could actually outperform an end-to-end training, which I will discuss later. Another very important and a common practice of whole-slide image analysis is to incorporate the information from multiscale, because as you can imagine, some of those features can actually span multiple scales. I mean, actually, this is really the case when the pathologists are looking for the features. They first look at the macro scale features. They look at what are the, for example, they're looking for neurons. They're looking for glands, which are like higher level higher level features compared to the individual cells, and then they localize the suspicious regions and then zoom in to look at the specific cell-based features. So, and for some cases, the optimal features can span different scales, and sometimes you have to combine the features that you look for at different scales in order to retrieve a accurate diagnosis result. So, and also another observation here is that, well, in the multiple instance learning, we do not consider any spatial relations of the patches, but in fact, those features are spatially continuous. So they actually, spatially clustered. So that is why for that purpose we utilize the feature pyramids in our solution. So basically we construct feature pyramids by concatenating the embeddings from the lower resolutions, which corresponds to a bigger scale, to a higher resolution, which corresponds to a finer scale. So basically what we do is that we simply duplicate the embeddings of the lower resolution, and then we propagate that embeddings to the higher level by concatenating that lower resolution embeddings to the higher resolution embedding, so that at the end, basically we duplicate that. So basically all of the image patches that form higher resolutions that belongs to the same lower resolution patch will get a copy of that embeddings from the lower resolution. So at the end, our embeddings will have features that span multiple scales. And more importantly is that if the patches are spatially close to each other, which means that they will belong to the same patch at a lower resolution, then they will have same parts in their embeddings. And then since we use the similarity calculation that we use here is simply it's based on the inner products, which means that if you have same parts in your feature vectors, then you will get potentially, the attention score will be potentially more similar because you have same parts in the feature vectors. And also another advantage is that the feature vectors now contains the information from multiscale. And this is the whole picture of the workflow. So basically we first extract the patches from different resolutions, and we train the embedder in a self-supervised learning. And after the embedder is trained, we compute the features and construct feature pyramids, and then fit the feature pyramid in the multiple instance learning model, and then train that multiple instance learning aggregator to form a prediction. And so the data set that we use a chameleon 16 data set, which is a lymph nodes, metastasis lymph node sites of breast cancer, which contains 400 slides. And the nice thing about this data set is that this data set actually comes with annotations. So we can, although in the training, we discard the annotations at training, but we can evaluate like the localization of how the attention scores actually corresponds to the localization of the tumor. And the other one is the one from National Cancer Institute. It's from a project called TCGA. And it's a lung cancer data set which contains two subtypes of lung cancer, the adenocarcinoma and squamous cell carcinoma. So this data set is fully unannotated. So we basically only evaluate the accuracy, slide level classification accuracy there. So and the result shows that so in the classification result, our model actually compared favorably to all of the recent models. And for localization result, although there is still some gap between the fully supervised model, but still we can see that still we can see that the result is promising. And if we were able to obtain a lot of like chameleon 16 unannotated slides, then we can see that the gap might potentially be met. And this is a result of the example of the attention map that's generated by the multiple instance learning models. And then we also evaluate the effect of using self-supervised contrastive learning here. So basically, we compare the two cases of the unbalanced bags and the balanced bag. And the reason why we pick these two data sets is that chameleon 16 is highly unbalanced, which means that in the positive slice, the tumor region only occupies a very small region. And for the TCGA data set, it's actually almost 80% of the regions actually contains the tumor. So we compare to the feature extractor that is learned from ImageNet, and also the the feature extractor that is learned from max pooling. So basically, as you can imagine, if you use max pooling, then you don't have to keep all of the patches in your memory because you only you can pull out the maximum instances dynamically, so you don't have to keep everything in memory. So for using that, you can also learn a feature extractor. And also, if you don't consider multiple instances at all, you just propagate all your slide level labels to all of the patches, you can still train a fully supervised, as you can still pretend that you're training a fully supervised model. And then that you can also get a feature extractor. So what we found is that the self-supervised contrastive learning gives you a lot of benefits when your data is highly unbalanced. So which means that the patch-based training will never work at all because you are propagating a lot of wrong labels to your positive slides, which contains a lot of negative patches. We also evaluated just the multiple instance learning aggregator that we proposed to the other recent multiple instance learning aggregators on some general multiple instance benchmark data set. So this data set contains a data set of conformations of molecules. Basically, the molecules can have different conformations, and only one of the conformation might be effective. So the goal here is to trying to see if any of the conformations of the molecule will even work at all. And the other data set contains like image fragments of different animals. And also the results shows that our model also achieved the state-of-the-art performance. And the summary so far, so we see that whole slide image classification can be cast as a multiple instance learning problem, relying only the slide level labels. And we proposed a new multiple instance aggregator with a critical instance and trainable measurements to other instances. And we see that self-supervised learning use robust features in weakly supervised learning circumstances, basically with only slide level annotations are available. So in the last few minutes, I wanted to basically try to discuss the problem with this multiple instance learning model in whole slide image analysis. Basically, we're trying to ask, this patch level modeling is it really good? Is it really good enough? So we know that the features might span multiple patches, and there is some sort of spatial relation of the features. Although we can have sort of solution based on we can make it better by using some feature pyramid, but it's not intrinsically comes in the multiple instance assumption. And also there is some more complicated that involve the separability of the multi-class problem. And I will give a simple example at the end. So first, we know that the patch representations cannot always capture enough information in many real tumor detection problems. So the data set that we use there, they are like the reason why it works there is because the cellular level features are enough to differentiate the negative versus the positive. Basically, you can look at the cell shape or the nuclei shape, or like if they're going some metastasis. If there is some sort of like cell level events that's been happening there. But for like for adenocarcinoma, and so basically when the tumors are the typical glands, the criteria can also include the abnormal position and the organization of ducts or glands. And the ducts and glands are a collection of cells that has a certain shape that can span multiple field of views. So basically, if you're looking at cellular level feature, you are not able to tell the whole picture of what the whole gland looks like. And sometimes the criterion can be if the gland happens to appear in some abnormal position, like whether it stays too close to a muscle bundle or it stays too close to a neuron. So these are like the features that would not be able to capture by the patch-based representation. So basically, we need to let the information flow between the instances in order to have a better modeling of the interaction of the patches. So that is recently there has been works on building graph neural networks on host-like image analysis. And one of the open questions here is that how to build an efficient graph from the input, just instead of just taking some image grids, some lattice graph, if there any way to build a more efficient graph out of it. It's basically built, so it's like an open question here. And also like there has been recent works on using vision transformer and positioning on host-like images, because they have like, with vision transformer, you have the ability to, for the pre-training of the feature extractor and also you can have like positional encoding spatially with your patches, so that your model can potentially learn some correlations that has the spatial information. And letting information flow between instances is important because the features can span more than an image patch. And also like we can also either explicitly or implicitly encode the position information by in the two models. And furthermore, it can generalize the multiple instancing assumption when the standard multiple instancing assumption is not appropriate. For example, we're looking at this simple example, last example here. So we're trying to, if we're trying to use multiple instant learning on classification of those images of beach, image of desert, ocean, and sky, we will partition this image into small patches. But then what we found is that there's actually not a single concept of a beach here, because a beach is consists of the sky, sea, and sand. And then the desert are the same. So you will have the sky and sand, and for the ocean, you have the sky and sea. And for the sky, you will have only sky. So basically, the problem here is that the instance level concept that you are seeing here with the labels is actually inseparable. So in this case, the standard multiple instance learning assumption will not hold. So that is why letting the information flow between instances will allow the hidden concepts, which is the actual concept that forms your label, the concept of your observed labels here. It's important. It's because and it will potentially allows greatly enable the model to have a greater capability to be able to solve some more challenging tasks. And that's really the end of the presentation. And I want to thank my advisor and the collaborators and all of the great lab mates and all of the sponsors. And I would like to take questions.
Awesome. I think that was a great talk, Vin. So I guess before we move on to questions, we can give a round of virtual applause. And yeah, I'll leave the floor open for questions.
Yeah.
May I ask a detail about the self-supervised training?
Sure.
How do you just pick one patch and then do random elimination on the same patch?
So that's actually a little bit. So that is almost true, but the really difference is the patch selection that what we decide to put in the same batch. So as you might know that for same clear, you will have to have a big batch size. So the image augmentation that we use is almost exactly same. It's exactly same as the same clear, but we carefully select what patches are selected to form the batch. Because as you can imagine, if you happen to select two patches that are cropped from adjacent regions, then there is no way to say, I mean, because in the self-supervised learning, you also crop two subregions from the same image. And if you are just fitting those images, that if you happen to fit two image patches that are that are adjacent to each other, or they're actually from a very similar region, then they are actually positive pairs. But in that case, it will be treated as negative pairs if you don't care about it in the training. Yeah, so that is the difference there.
So for negative pairs, do you sample patches that are far away?
Exactly, like patches that are, yes, yeah.
Okay, got it, thanks.
And do you do any kind of filtering of the patches even beforehand, just to avoid the smudging and the corner issue?
Yes. So basically, like one basic way is to get rid of the backgrounds, which are basically blanks, and that gets rid of like about 50% of the patches. And then there is also some prior knowledge about it. Like if, for example, mostly we know about, because the very nature of the staining, we are not caring about, for example, we're not caring about blood cells, then we can select that hue like with red and then get rid of all of those. And that will decrease, and that will make the model to converge even faster and will decrease the computational cost. Yeah.
So during the patch selection also, you already did this all filter before when you are creating these pairs for contrastive learning, right?
Yes, exactly.
Okay.
Yeah.
Okay. And do you have any results about the contrastive learning only, like how the pairs looks like finally, and did that make any kind of improvement?
Yeah, we actually so what we do is that we do some sort of like data retrieval tasks here. Like we select the most similar patches based on the self-supervised features. And actually, we actually did that experiment. It actually gives some really good result on that. I mean, almost give you very similar patches from it. So we think that, I mean, that is evidence of the
right.
reputations are actually good enough. But we also found some problem with those. It's like they are not really quite, like the dynamic range of the differences are actually pretty low. So you might have the top match, you will have something like 0.9 something, but for the very dislike patches, you will still have something like 0.8.
Wow.
Yeah, so certainly there's a lot of improvement there.
So did you see, like during your retrieval, did it also try to identify any patches outside the same sample? Like did it identify some like those similar patches?
It's actually we are actually trying to determine the similar samples from the whole data set.
Whole data set, okay.
Yeah.
I have a quick question. Actually, we're kind of running out of time. So if there are other questions, I'll let other people ask. Otherwise, I can ask one question. Okay, so one question was, I found this multiple magnifications part to be very interesting. And as you said in the end, like the advantage of the vision transformer or the graph gives you a nice way of getting this positioning based, the spatial relationship. I was wondering if you could also just use multiple resolutions to get the same thing. So for instance, you have this 5x and 20x magnifications, but if you have like a 5x, 10x, 15x, and 20x, wouldn't it get features from like multiple resolutions, which
you can have features of multiple resolutions, but you then, for example, there will be a same question for each resolution though. For example, in your 5x resolution, do you really care about the information of adjacent patches at all? You cover all of the patches that belong, all of the patches from a higher resolution that belongs to the lower resolution. But then what about all of the adjacent patches in the same lower resolution? So it's actually a orthogonal considerations there. So you can consider both, actually. I would say it will be beneficial if you consider both. And also you can even treat the lower resolution image as some sort of like mega nodes in your graph models. And that will be more flexible, I think.
Yeah, I think that's a great idea. Thank you.
Yeah.
Are there any other questions? Okay, so if not, thanks so much, Bin, and we'll have the talk uploaded on YouTube. So if others have follow-up questions, we'll put them in touch with you.
Sounds good.
Yeah, and